Good morning, class. Today I will be starting with a new chapter, Origin of Life. Now, life is that characteristic which an organism possesses to maintain itself and to produce more of its own kind, that is, for reproduction. Origin of life on this earth has always been a puzzling problem since ages, and diverse views have been proposed for origin of life on earth, and these views are: the first one is theory of special creation. According to this theory, it is believed that some supernatural power, that is God, has created life on earth. According to Christian mythology, it is believed that life on earth was created in 6 days, and on the 6th day the first man, Adam, was created, and then the woman, Eve, was created. According to Hindu mythology, it is believed that Lord Brahma created life. So this is one theory called theory of special creation. After that, the second theory proposed is theory of spontaneous generation, or abiogenesis or autogenesis theory. According to this theory, it is believed that life has been created from abiotic or non-living matter, and it is believed that the warmth of the sun, the air, different non-living elements, moisture, all these together created life on earth. And this view of creation of life by theory of spontaneous generation was supported by scientists like Aristotle, Epicurus, and von Helmont. Then this theory was not successful. After that came the theory of biogenesis. Now, this theory of spontaneous generation was discarded by Francesco Redi, by Lazzaro Spallanzani, and by Louis Pasteur. They said that life can originate only from pre-existing life, and they performed various experiments, and by that they proved that life cannot be generated or cannot originate spontaneously from abiotic matter. There has to be pre-existing life for creation of life. So the experiments done by them, Francesco Redi, he took boiled meat in 3 flasks, and 1 flask he kept that open, and the other 2 flasks he covered. The flask, one with muslin cloth, and the second flask he covered with parchment paper. And he saw that the flask that was uncovered, some bacteria, some worms, some insects were seen on the meat pieces. Whereas the 2 flasks that were covered, nothing grew inside the flask on the meat pieces. Only they were eggs and larvae of insects on the surface of the muslin cloth or on the parchment paper. So he concluded that life can originate only from pre-existing cells. Then Spallanzani, he did an experiment. He took a flask with nutrient medium, with boiled nutrient medium, and sealed the flask from top, and saw that there was no growth inside the flask. Then he cut the mouth of the flask and left it open, and he saw that there was growth inside the nutrient medium. So he concluded that it was through air that some form of life had entered the flask, and on nutrient medium more cells could be produced from pre-existing cells. Same way, experiments done by Louis Pasteur. He also took yeast and sugar solution in 1 flask, and he heated the top of the flask and bent the tube into an S-shaped structure. So S-shaped structure, when air entered it at this point where S was formed, the air was trapped at that point. 1 minute, I'll get the pen and explain it to you. See, Louis Pasteur had a flask like this, and the mouth of the flask he heated and made it soft and made an S-shaped structure. So this flask here had yeast and sugar solution, and air that entered from here, all the organisms or the spores or any other form of life, they got trapped here. They did not reach the yeast and sugar solution, so nothing grew in the flask. Then when he broke this part, directly air could enter the medium and he saw growth over here. So based on the experiments conducted by all the three, that is Francisco Redi, Lazzaro Spallanzani and Louis Pasteur, they all said that life originates from pre-existing life. Then the fourth theory proposed was biochemical origin of life. This was given, experiments were done by Oparin in the year 1924 and Haldane in the year 1929, and independently these two scientists studied life, and this is the modern theory for the origin of life, and so far this is the most accepted theory of origin of life on earth. And Oparin also wrote a book called Magnum Opus, or Origin of Life, and they said that life was a gradual, origin of life was a gradual process. Life did not come into existence suddenly, but it was a result of a long series of physico-chemical changes. These changes brought about gradual evolution, first of certain inorganic compounds and then organic compounds. Then the whole series of events which they have described in Origin of Life can be divided into 8 steps. So now I will be telling you about the most accepted theory for the origin of life, that is biochemical origin of life, or Oparin-Haldane theory, and the 8 steps for the origin of life. First is the origin of earth. We are talking of Oparin-Haldane theory, which is also called biochemical origin of life. The 8 steps described are: first is origin of earth. This is the Big Bang theory. Now, it is believed that the earth originated 4.5 to 5 million years ago due to a part which was broken off from the sun. That's how earth came into existence. Then second step is primitive atmosphere that was created on the earth. This origin of earth is believed 4.5 to 5 million years ago. Now, a primitive atmosphere, which means the origin of molecules and simple inorganic compounds. The temperature of the earth at that time was very high. It was believed to be 5,000 to 6,000 degrees Celsius. Elements like hydrogen, oxygen, carbon and nitrogen could not exist in free state. They combined among themselves or with metals, forming oxides, carbides, and nitrides. These compounds were present in gaseous state, and water was present as superheated steam. All these compounds together formed the primitive atmosphere. And the atmosphere of the earth was supposed to be reducing. Atmosphere of earth at that time was reducing and not oxidizing as it is today. Today we have an oxidizing atmosphere, but at that time the atmosphere was reducing and not oxidizing as of today. Then third step was formation of simple and complex molecules. Formation of simple and complex molecules. This is the third step proposed by Oparin and Haldane. As the atmosphere cooled down to 1000 degrees Celsius, various saturated and unsaturated hydrocarbons were formed. Gases like ammonia, methane, carbon dioxide, and compounds like cyanides, phosphate, etc. were formed. These compounds came to earth along with rainwater and resulted in the formation of more complex organic compounds in oceanic water. The oceanic water was described as hot, dilute soup. So in the third step, the oceanic water was described as hot, dilute soup because it contained lots of organic and inorganic compounds. Then the fourth step was synthesis of complex organic compounds. Complex organic compounds. This was the fourth step. Ultraviolet rays, cosmic rays, and electric discharge stimulated the simple organic compounds to interact. This interaction caused them to combine and polymerize to form complex organic compounds such as polysaccharides, nucleotides, lipids, proteins, and nucleic acids. The fifth step was origin of colloids. Fifth step was origin of colloids, coacervates, and individuality. This was the fifth step. The organic macromolecules, due to intermolecular attraction, aggregated into various combinations, and then they precipitated out in the aqueous medium in the form of large colloidal particles which were known as coacervates. So coacervates are large colloidal particles. Coacervates were a mixture of large proteins and polysaccharides with some water. They did not have any limiting membrane of lipids. They showed little metabolism. Coacervates have been looked upon as precursors of the first organisms, that is, the prebiotic structures. Coacervates were later surrounded by a limiting membrane which was formed by the organized molecules of fatty acids like lecithin and cephalin. They also started absorbing organic substances from oceanic soup. They then turned into anaerobic heterotrophs. Then the sixth step for origin of life is origin of autocatalytic systems, genes, viruses, and primordial life. This is the sixth step. Origin of autocatalytic systems, genes, viruses, and primordial life. The first living cell, nucleoproteins, appeared as self-duplicating, so were capable of performing hereditary function. The first sign of self-perpetuating life was thus displayed by the nucleic acids. The monomer nucleotide units of nucleic acids were comparable to the present day genes. A number of such genes might have aggregated into a large unit called protovirus, which is compared to the present day virus. These were probably the first, most primitive or primordial organisms. The rise of viruses. They were the first and the most primitive or primordial organisms. The first organisms were heterotrophic, they were anaerobic and virus-like nucleoprotein structures. For energy, it was dependent on complex organic compounds from seawater, hence they were also called chemoheterotrophs. Chemosynthesis means the formation of complex compounds and nutrients from inorganic or simple organic compounds within the cell itself. Hence, the chemoheterotrophs became the chemoautotrophs, and sulfur bacteria, nitrifying bacteria, and iron bacteria belong to this category. Then the seventh step is origin of photosynthesis and production of free oxygen. This was the seventh step of operant held in theory, or the theory of biochemical formation of life. After some time, certain chlorophyll-like pigments were developed. The organisms which developed such pigments could utilize the sunlight to form nutrients. These were then called photoautotrophs. They made use of water as a source of hydrogen and liberated free oxygen, and they were called aerobic photoautotrophs. These photoautotrophs evolved about 3,500 million years ago. Photoautotrophs that could utilize energy of the sun and produce free oxygen. They came to Earth about 3,500 million years ago. And these organisms resemble the present day cyanobacteria, that is the blue-green algae. Eighth step of origin of life is origin of the eukaryotic cell, those that have a well-defined nucleus. This is the last step, or the eighth step of Operin-Haldane theory. Liberation of free oxygen into the primitive atmosphere by blue-green algae like prokaryotes was a revolutionary change in the early history of Earth. The free oxygen oxidized methane and ammonia, forming carbon dioxide, nitrogen, and water, and the reducing environment then changed to oxidizing environment. The free oxygen formed a layer of ozone above sunlight, thereby blocking the ultraviolet rays of the sun and making the organisms to migrate on the land from the water. Conditions became suitable for aerobic respiration. About 27 billion years ago, the prokaryotes were gradually developed a true nucleus, mitochondria, and thus free-living eukaryotic organisms originated in the primitive ocean about 15 billion years ago. So, how did life finally turn into eukaryotic organisms? Is a long series of changes. We have seen in this theory long series of changes that are physico-chemical in nature. Took place for evolution of life on earth, for origin of life on earth, from very simple inorganic compounds and organic compounds to complex compounds to colloidal substances, finally production of the first form of life, which was prokaryotic in nature, and how membrane developed around the nucleoproteins and a eukaryotic cell was formed. So this is a series of changes. It cannot occur in just one day. It took several years, several million years, because Earth originated as a part of sun. Part of sun broke, temperature was reduced. Finally, it became suitable for life to exist on Earth, and that's how a single eukaryotic cell originated on Earth. Now, earlier we saw that the primitive atmosphere was such that it was reducing, whereas today's atmosphere is oxidizing. Now, oxygen has been produced by photosynthesis. So now what we have is an oxidizing atmosphere, unlike earlier times. Now we are going to study the effect of oxygen, how oxygen has affected life on Earth. Reducing atmosphere is essential for abiotic synthesis. Atmosphere on the primordial Earth was a reducing one, as there was no free oxygen. There was an abundance of reducing gases such as hydrogen and ammonia. Due to the absence of free oxygen, burning and oxidation of substances was not possible. These conditions were ideal for the formation of compounds necessary for the origin of life. Moreover, free oxygen would have wiped out most of the anaerobic protobionts, which later evolved into true cells. With the evolution of oxygen, free molecular oxygen brought remarkable changes in the primitive atmospheric conditions. The Earth's atmosphere changed to oxidizing atmosphere. The methane and ammonia began to disappear due to oxidation by free oxygen. Methane changed into carbon dioxide and water, and ammonia changed into nitrogen and water. As ozone layer developed, it helped in preventing the entry of ultraviolet rays to the Earth. Now, with the appearance of oxygen in the primitive atmosphere, the process of origin of new life forms stopped because free oxygen oxidized most of the intermediate products. So, we can to some extent say that reducing atmosphere is a necessity for the origin of abiotic life. Now, Liederberg summarized the entire process of origin of life in 3 major steps. Liederberg summarized life in 3 basic steps. First is chemogeny, Lederberg. First step is chemogeny. Second step is biogeny. And the third step is cognogeny. Now, one by one, I will tell you what chemogeny means. It is the phase in which chemical evolution took place, resulting in the formation of simple chemicals. That is, phase of chemical evolution, where simple chemicals were formed. And biogeny is generation of life. Biogeny refers to the origin of first living cell. Origin of first living cell. The prokaryotes, virus-like cells that were produced. And cognogeny means generation of complexities that arose. So this refers to the development of complexities in the form and function of living organisms. Development of complexities in the form and structure of living cells. So this is how Lederberg has summarized the entire process of origin of life. Oper and Haldane had described these 3 simple steps into 8 steps. How Earth originated, how primitive atmosphere came into existence, then simple molecules were formed, complex molecules were formed, Earth's atmosphere became reducing, then how nucleic acids, nucleotides were formed, nucleic acids were formed, how viruses and prokaryotic cells came into existence, then how photosynthesis occurred, oxygen was produced, and eukaryotic cells were produced. This they have described in 3 simple steps: formation of chemicals, origin of life, and origin of complexities in structure and in form of life. So now one very important experiment which proves Oper and Haldane theory is the Eure-Miller experiment. Now, in Origin of Life, given by Oper and Haldane, the 4th step, which says synthesis of complex organic compounds by means of UV rays, cosmic rays, electric discharge. They said that the simple organic compounds interacted, and this interaction helped to polymerize these simple compounds into complex compounds such as polysaccharides, nucleotides, lipids, proteins, and nucleic acids. This is what Eure and Miller proved in their experiment. So this is Eure-Miller experiment is a proof. It says Eure and Miller successfully synthesized amino acids and some complex organic compounds in artificially created primitive atmospheric conditions. They created the atmosphere containing hydrogen, primitive methane, and ammonia in a flask. Water vapors were allowed to enter in this flask through a side tube. Energy was given by heating the liquids and also by electric sparks in gaseous chambers. The experiment was conducted for several days. The turbid liquid present in the trap was analyzed and they were able to identify amino acids like glycine, alanine, etc., and a large number of complex organic compounds like hydrogen cyanide, adenine, and other nitrogenous bases in the contents. Such a synthesis in the absence of oxygen and living organisms was called abiotic synthesis. This type of synthesis cannot occur in the present day oxidizing atmosphere because free oxygen would destroy most of the intermediate products. So this experiment clearly explains the necessity of a reducing atmosphere for abiotic synthesis. And the apparatus which Eure Miller used, I will draw that diagram and you will see how in a reducing atmosphere in the absence of oxygen they could create complex organic substances. They took a glass mug, this is the condenser. They had two electrodes, positive, negative. Sparks were created here. Sparks were created here, and this is a condenser. Now, because of the sparks, methane, ammonia, water vapor, hydrogen, etc. were produced. Now, this is bringing steam. They had a flask here in which they were boiling steam. This was the trap. This is the direction of movement. There is water here. So steam entered through this here. This was a mixture of gases present here. By electric sparks produced, these are the electrodes, positive electrode and negative electrode, anode and cathode. Sparks were produced, these gases were formed, condensation took place here, they moved here. This is the trap. Now, when analysis of compounds in the trap was done, it was seen in the trap that synthesis of complex compounds like nucleotides, cyanides, and other compounds like nitrogenous bases and all were seen here. Now you see, all this formation is taking place in the absence of oxygen. So he said that a reducing atmosphere was needed. Eure and Miller said that a reducing atmosphere was needed for abiotic synthesis, as was proposed by Oparin and Haldane in their theory of biogenesis for origin of life. That is why theory of biogenesis so far is the most accepted theory for origin of life on earth. So this much is enough for today. I will stop today's class here. Take care and good luck to all of you.